about elements. Ready?
Periodic table.
Yep. Periodic table. So most important elements in living organisms. 4 big ones. Carbon, hydrogen, oxygen, and nitrogen. It says here, hydrogen is always written as H2. What that means is if you have hydrogen by itself, it's always 2 hydrogen atoms bonded together. You may find H with a different subscript, like a chemical formula. Like methane is CH4. That's okay because it's not pure hydrogen. If it's just pure hydrogen, just H by itself, it will always be H2. Same with O2 and N2. So then, to make a molecule, we put 2 or more atoms together. And then, if we have at least 2 different types of elements, we have a compound. So every element on the periodic table has its own atomic symbol. And these symbols always start with a capital letter. Some elements have 1 or 2 lowercase letters after the capital. Usually the only ones that have 2 lowercase are the ones that haven't gotten a formal name yet. They're given placeholder names, or basically they take the atomic number and turn it into Latin words. So like atomic atom number 118 right now is known as ununactio. So it's UUO. Once it gets a name, it'll just have the regular 2 letters. So to find out how many different elements we have in a molecule, simple. Just count the capital letters. How many elements are in N2?
Huh?
1. How about CO?
3?
How
No.
many capital letters do you see in CO?
Oh, wait, never mind. I was thinking of CO2.
Well, even in CO2, how many capital letters do you see?
Oh, wait, never mind. I'm
It's
tired.
not asking how many atoms, it's asking how many different elements.
Oh,
You're right, there would be 3 atoms, but only 2 elements.
okay.
How about NaOH? That'd be 3. All right. So that's just a little bit of background chemistry. And then we're going to switch over to the building blocks activity. Same assignment, other document. And we're going to look at the building blocks of life. Molecules that these atoms are put together that basically let us exist. For each of these types of molecules, there's a poster. And you can look at this and find the information you need to answer the questions. So, are there any questions around what you need to do here? We have 20 minutes on this, and then we're going to go over it together. Time is yours. Go ahead and get started.
Thank you.
Grilling with Ace. Get a new grill and choose from
What are we listening to today?
What do you think?
Either Disney music or Christmas music.
Disney?
I've heard Disney. And The Greatest Showman. You can't lie. I've heard it.
See participating stores for scheduling or exclusions.
Oh, I'm not ready for Christmas. Bless you.
It goes like this, the 4th, the 5th, the minor fall, the major lift, the baffled king,
Yeah.
And if you can't find absolutely everything, that's okay. That's what we're going to go over it.
I think I died halfway down the hall. I'm not actually here. It's a hallucination.
That's okay.
This is all a figment of my imagination.
Then I can maybe eat more frequency if I'm hallucinating.
There's a blaze of light in every word. It doesn't matter what you heard. The holy or the broken.
Yo, this song slams. I'm either gonna be hysterical or on like really tired. That's how it's going today.
Did you do anything with the test yesterday?
Not yet. I got like one question done. Can you? All right, you want me to do that first?
No.
Oh,
I forgot that I reopened it. Which one yes.
okay. This was a very unsteady desk. I should probably just turn up my brightness, but I don't want to.
Wendy's new classic chicken sandwich is now in the 2 for $5, and that's reason to celebrate. Try the new classic and then take your out for a victory lap with the iconic Dave single, the delicious spicy chicken sandwich, spicy or crispy 10-piece nuggets, or just get another classic chicken sandwich. Taste greatness today with Wendy's 2 for $5. We got you.
Can I listen to music?
You survive the demands of distance learning without crying your eyes out. That's vision accomplished. Whatever your goal, BSC vision coverage always be free.
How do you know that?
Enroll or afford Bioplan. Learn more at bsc.com.
Yeah. How do you know we didn't cry? You better watch out. You better watch out. That's it. I hate that song. It's creepy. Oh, this isn't worse. Someone hit me upside the head with a baseball bat as hard as they can. Put me out of my misery. Just shakes head in disappointment.
One December, I shall see a world and not free. Tell me no more. When we learn what Christmas is for. When we found what life really worth, there'll be peace on earth. Someday all our dreams will come to be. Someday in a world where men are free. Maybe not a dime for you and me, but someday Christmas time. Someday Christmas will be there, with no hungry children, no empty hands. One happy morning, people will share a world where people care. At Christmas, there'll be no tears. When all their evil has none has fear. One shining moment was spread away from our world today. Someday all our people come to be. Someday in a world where people are free. Maybe not a
Is this pentatonics or whatever? I love this band. It is.
Before you drive your own Nissan Rogue, you gotta ask yourself, how rogue are you gonna go? We talking kind of scary animal desert road, no igloo in the middle of nowhere road, or take a long way home just because kind of road? Just a question, but with top available drive modes, you're sure to find the answer. Go rogue in the all-new Here's to Reimagine 2021 Nissan Rogue, now with the most standard safety features in its class. Continue, Miss Bonner.
Unlimited double miles on every purchase every day.
Objection! My credit card doesn't say miles at every purchase.
Reject your objection. With the Capital One Rent-A-Car, you earn unlimited double miles on every purchase. Order. And right now, earn 100,000 bonus miles when you spend $20,000 your first year. I don't know. No further questions, Your Honor. Because what's in your wallet?
Limited time offer, terms apply. See CapitalOne.com for details.
looks like a lot of people are ready to go over this. All right. So what were kind of the important topics, the important things here about these different carbohydrates and sugars? So glucose. Where does glucose come from?
Sugar. I'm kidding.
It
Food.
is a sugar.
Food.
How does glucose get made?
No, it's like
Yeah, this is the product of photosynthesis. This is the plant making its energy to live off.
okay.
The other thing important to notice about glucose is it's a small molecule. It's easy to move around, easy to store, easy to break down. Starch. What is starch? Debbie.
What plants use to build two different linear polymers?
Well, starch is one of those polymers. And glucose is what makes up the starch. It's just a whole bunch of glucose molecules strung in a chain together. And this is one of the ways plants store their sugar for later use. What is a really starchy food you can think of?
Potatoes.
Pasta. Okay, potatoes. Pasta, that starch comes from the wheat, where it stored those sugars for the new sprout that's going to germinate from that wheat seed to use to grow until it can do its own photosynthesis. Potatoes, same idea. They store the starch underground in the potato, so then in the spring, when they have to push up new shoots, they can do that with the energy they stored in the starch. Now, cellulose. This is the other long-chain polymer plants make. This one we can't use. Starch we can digest. Cellulose we can't. Where is cellulose found in plants? Do you know that? Brady?
Stem, right?
Well, yeah. It's in the cell wall. Basically, it's in every cell wall of every plant. That's what makes the table able to stand up. Is the cellulose in there? There's enough of it to give it that hard rigidness. So this is for energy. This is for structure. That's the big difference here. Glycogen. Considering our bodies can't make either of these, we have to do something when you eat that Snickers bar and have all that extra glucose floating through your system. So what our cells do is they take up the glucose and convert it to glycogen. Then when your blood sugar drops, that glucose gets re-released into the bloodstream to try to keep you kind of in that normal range. So, lipids and cells. So, phospholipids are a pretty special kind of molecule. They're kind of weird because one portion of it is a long water-fearing or hydrophobic carbon tail. And the other part is a water-loving or hydrophilic phosphate. So what do those phospholipids make up in our bodies? What do they form? Sean?
The lipid bilayer.
The bilayer? Yeah. That is in fact our cell membrane. So what happens is we've got these little phospholipids floating around out here, and they're kind of like oil. What happens when you mix oil and water?
They don't mix.
And they make those little circles, right? So those circles come because the oil goes with another oil molecule, and then another oil molecule, and they make this big solid wall of all the tails so that water can't get in there, and then all the other oil molecules gather up in there. This is kind of the same idea, except we have this part of the molecule that really likes water. So what that does is it actually orients it so that it's either pointing that way or that way. Because this part has to be touching water. These parts get all tangled up like this and make a nice solid watertight seal. So when molecules want to leave the cell, if they try to go between the phospholipids, they're going to have a hard time because this is pretty full of lipid tails. So then they have to go out through a channel protein that your cells can regulate. We're going to look a lot more at this when we get into the cells here. So hydrophobic means water-fearing, or it doesn't want to have anything to do with water. What else do our bodies use fat for besides the cell membrane?
Energy,
Energy, long-term energy storage, yep. What else?
body heat.
Insulation, yeah. The fat doesn't actually generate the body heat, it traps body heat.
Well, yeah.
All right, last one here, DNA and nucleotides. So we got 4 nucleotide bases. So what are the 4 bases here? Meredith.
Adrian and thymine. Cytosine.
Yep, adenine, thymine, cytosine, and guanine. Then hooked to one of those, we have a deoxyribose sugar. This is where the D in DNA comes from, is the deoxyribose. And we have a phosphate group. With those 3 things together, we have what's called a nucleotide. So, how is an RNA nucleotide different from a DNA nucleotide? 2 ways. Sean.
The deoregulatory is replaced by the ribose.
Right, instead of deoxyribose, we just have regular ribose, which is why this is RNA for the ribose. And what's the other one, Grady?
Thymine base is replaced by uracil.
Right, T is replaced by U. And the result of that, because they use a different sugar, the way the molecule can bend means it can't be double-stranded. It has to stay single. Now, with DNA, when it is double-stranded, forms a double helix of complementary strands. What that means is that each nucleotide has a specific partner to match up with. Look at this diagram. How do you think they know which partner to match up with? Brady.
Someone on their side of the helix.
Well, it's not, they have to match up with a specific letter on the other side of the helix. Look at the AT pair and the GC pair. What's different?
G go. Sorry. G goes with C, and C always goes with a G.
Okay, so but I mean, if we look at this right here versus this right here,
Yeah.
what do you see, Brady?
The A and T only has like one of those shapes, like that. That line's not closed off. Is that it?
Yeah, this only has 2 of these little hydrogen bonds. This one has 3. That means that if C is trying to bond, it's looking for something with 3 spots to accept the hydrogen bond. So if it finds T or A, it's only going to find 2, so it's going to move on and look for C. Does that make sense?
Try it one more time.
So basically, these guys need to form 3 hydrogen bonds.
Yeah.
So if C tries to match with T can only form 2.
Yeah.
So it's going to have one here that's still unhappy.
Oh, okay. That's a funny way of putting it. It's going to be unhappy.
Oh. This also means the AT bond is weaker,
Okay.
which when we get to genetics actually becomes important in determining where we start copying proteins and DNA.
I actually learned a lot about. I love genetics. Is that weird? I love learning about them. It was
So-
one of my favorite things.
A always pairs with T, C always pairs with G. What that means is that you can use one strand as a template for the other strand. A template meaning that you can just use it to make another copy, like a blueprint. So in 2001, this is when something known as the Human Genome Project finished up. Human Genome Project was started in 1998 as an effort to sequence the entire DNA of a human inside of 10 years. And clearly they succeeded because they got it done in about 3.5. And nowadays, gene sequencing has gotten so fast, you could do an entire human's genome in 3 days. And this opens up a lot of stuff for us medically. Because if you know the genetic cause of a disease, the specific genes that changed, you can do a couple things. One, you can do testing for those, like before somebody even has a child, you can test to see if they have the genes for something. Two, there's technology called CRISPR, C-R-I-S-P-R. And what it is it's a way of editing DNA, where you can go in and change small segments or individual bases. This hasn't been done in humans yet, as far as I know. But it opens up the possibility of using that to cure genetic diseases like Huntington's disease.
I know someone with that.
Or cystic fibrosis, any of those, sickle cell anemia. Those are all genetic diseases you might be able to cure. So.
What about ALS? Or is that
ALS,
genetic?
There's 2 forms of ALS.
Yeah.
The familial form, you could cure that. The other form, I don't know if you could.
Okay.
There's some evidence that there's an environmental aspect in that, but I don't know if that's also linked to a genetic mutation.
All right. Sorry, I was curious.
No, it's okay. Good question. So, okay. So the idea here, let's talk about the building blocks here. Carbohydrates, glucose is what they call a simple sugar, 1 ring. It's the building block of these complex carbs, your starches, your celluloses, your glycogens. So they're just a bunch of glucoses all stuck together. And the specific way you stick them together determines the structure of the molecule you're going to make. Lipids, your building blocks are these fatty acids, the carbon tail, and then the backbone of it is a glycerol. We're going to look more at that on Monday. And for DNA, the building block is a nucleotide. When you put all those together, you get a DNA molecule. All right, we got about 3 minutes left. Are there questions on what we went over today? Okay. So, what I would like you guys to do is think for a minute from the first quarter's over. So think to yourself about what's going well and what could be fixed. I'm not talking about necessarily what you're doing, like what's working for you in class and what's not. And I want you to think about that, and I'm going to put up an assignment where you guys can post your responses to that. So I want to get your feedback so that we can, this is all new to everybody. So I'm trying to make this more engaging, more workable, less.
I think you already posted it.
What's that?
I think you already posted it.
I don't think.
What is the? Oh, never mind. That was the wrong thing. It was.
You were looking at the work completion survey?
Yeah.
Yeah, I'll probably be posting that survey tonight at some point. So. Anybody got plans for the weekend?
Sleep.
Always a good plan for the weekend.
Yep.
Anybody going out of town for Thanksgiving?
Nah.
Okay, good.
Just going to my grandma's house.
I was really hoping none of you were going to have to quarantine for 2 weeks when you got back.
Yeah. I told my mom, I'm like, we could just take a few days off. She goes, Ellie, no. She goes, nice try.
All right, I'll see you guys on Monday.
Goodbye.